So it is my pleasure to introduce our first speaker for the symposium, who is Marom Bikson, who is a Harold James Professor of Biomedical Engineering at the City College of New York, of the City University of New York. The translational research and development activity of this group spans preclinical studies, computational methods, device design and fabrication, regulatory activities, and clinical trials. And they develop a lot of technologies, obviously, and many of those are in clinical trials in over 350 medical centers. Dr. Bikson has published over 400 papers and book chapters and is an inventor on over 30 patent applications. So it's my pleasure to introduce you today, and I very much look forward to your talk.
Great. So thank you for that introduction. I want to thank the organizers for the opportunity to be here at this amazing venue. I also want to say, and this is like a little bit emotional for me, is I'm in New York now, but I grew up in Brookline. And I was looking, and if it wasn't for some of those trees, I could see my house from here. So that's really nice. I'm going to be talking to you about something that we've been working on in my lab for over 7 years now. But only recently did we get it to work. And this is this notion of this concept of wearable disposable electrotherapy. And these are my disclosures. So I'm going to be talking about electrotherapy. I'm going to be talking a lot about brain stimulation, neuromodulation. These are terms maybe many of you are already familiar with. And there's a whole class of technologies in this space. Some of them are implants, things like deep brain stimulation, spinal cord stimulation. Some of them are risky enough or expensive enough that you would need to get them at a hospital. These would be things like ECT or transcranial magnetic stimulation. And some would fall more into this category of wearable technology. So these would be battery-powered technologies that you could wear on your belt or a TENS device, right, that you could get at the CVS around the corner. Despite all the work that's been done on brain stimulation and neuromodulation, and there has been really decades of amazing work, major advancements in research and in clinical domains, it's obvious to everyone that electrical therapy pales in comparison to drug therapy, right? You go to your doctor, something's wrong, they're not going to hand you a battery, right? They're going to hand you a pill. And so this is a question that interests me as to why that is the case. You know, and one simple answer is that, oh, the brain stimulation doesn't work that well, right? Or that it's associated with more side effects than drugs. And I don't think that's the case. Brain stimulation side effects are often very mild. They're things like itching, right, compared to what you get with drugs. And I don't think it's the efficacy either. So a lot of thought has been put by me and other people into this. And I think the real difference actually comes down to distribution. It comes to how we access it. And so that's really going to be the theme that I want to talk about today. And I think it's very well aligned with this conference. So let's talk about maybe a typical patient journey and how they might or may not eventually get access to something like brain stimulation, right, at a TMS clinic right here in Boston. So obviously, the healthcare journey usually starts with our primary care provider. They are really the front line of medicine. And actually, most drugs are prescribed by your PCP, right, not by specialists. So let's say you are depressed, you might go through one or multiple rounds, right, of pharmaceutical treatment. This could go on for months or even years with multiple cycles. And only if you remain refractory, right, or you're not responsive, you have unacceptable side effects, are you then going to be referred to a specialist, psychiatrist, neurologist, and then possibly more rounds of pharmacotherapy. Maybe you'll be referred to a specialist who knows about ECT and TMS, and then you might have access to it. Now, this is associated already with a high level of burden. We're talking about daily treatments going on over many weeks, right? So you can imagine it's Boston, it's January, you're sick as it is, you got to drive 45 minutes through traffic and pay for parking for a 20-minute session, and then you got to go back in your car, and you have to repeat this day after day. So there is a burden right there of access related to distribution. Only the most refractory patients, right, will then be referred to surgery. This could be ablation, or it could be functional neurosurgery. So now you're getting your DBS, now you're getting SES, right? So the way I've drawn it here is brain stimulation and neuromodulation is really at the end of this cycle. And so the question is, well, can brain stimulation be a first-line therapy? Can it even be something that your PCP, they do hand you a battery after all, right? Instead of a pill, right? And they say, here's this battery. The side effects are very mild. You can use it in the comfort of your own home, where there's going to be much stigma, right? And so this is wearable medicine as opposed to ingestible medicine. Now, I already mentioned these technologies exist to some extent already. I and other people in this room have actually been working on them for many decades. But I feel they remain fundamental barriers to this, which is why your PCP is not handing you a battery. And this is what the experience is for a typical home-based, right, neuromodulation device. You have an initial purchase of the device, it comes hardware, whatever the packaging, batteries, maybe it's rechargeable cables, electrodes. These are accessories that have to be connected every single time, right? So you got your device, you have to connect the cables, you got to put it on your head, you got to make sure you've done it right. So you're futzing with it. It is, in theory, portable, but it's not the kind of thing you're going to carry in the back of your pocket. Every time you use it, you need to program it, right? What is intensity? What is frequency, right? And so on. And then there's waste associated not just with the accessories, but with the final sort of discharge and landfilling of the product. And if you look at a lot of research in the space, including from your own lab, it actually tends to focus on more complexity, right? It's adding sensors, it's adding AI, right? Which is all wonderful. But we suggested an alternative vision. So imagine if instead electrotherapy was available like a pill, or like a topical cream, or like a Band-Aid. And you would go perhaps over the counter and you would get a box, and that box would be indication specific, right? The depression Band-Aids, the pain Band-Aids, the movement disorder Band-Aids. And these would be distributed in individual strips, just like a Band-Aid. And you would take an individual strip, and that strip would be a dose. Just like a pill is a dose, the strip is the dose. And that would come with a prescription, like take one in the morning, one in the afternoon. You could carry it with you, fold it in your pocket very discreetly, apply it as needed and when needed. It would automatically provide a single dose, and when it's done, you would throw it out. And you can see how the bottom traces is really very akin to how pharmacotherapy would be distributed and used, right? So that was the broad vision, and that's what we're really thinking about in that way. And I think once we have a technology like this, it is quite possible for brain stimulation to become a frontline therapy, an alternative to pharmacotherapy, and this is what we call wearable disposable electrotherapy. So this is a technology here. Brought some with me. So this is a link to the preprint, and hopefully the Nature Communications paper will be appearing any day now. It's a pouch, and everything here is inside the pouch, right? It's a Band-Aid. It's less than 1 millimeter thick. In fact, most of the thickness comes from the packaging itself. This is a single dose of electrotherapy. All right, and so I want to describe to you how this works and how it can be used both for brain stimulation, but also for accelerated wound healing, and also for transdermal drug delivery. So with any engineering project, we started with our specifications, our design inputs, and we set the bar, I think, pretty high. We wanted a trivial user experience. What that means is not only is there no frequency or intensity knob, there's no on-off switch. There's no connections. There's no components. You simply apply it, and it turns on, so no settings. We needed it. This is going to be a single-use device, so we weren't obviously comfortable making something that would be environmentally irresponsible, right? So we focused a lot from the beginning on only selecting materials that are environmentally benign, as well as making the decision that we wanted to do this without electronics. So for those of you who are generally familiar with the engineering of stimulation devices, what we're talking about here is current controlled stimulation with a prescribed duration, a current controlled intensity, a prescribed waveform, right, without electronics. And this was a very odd proposition, and I get a lot of questions of this from the engineering audience. From the beginning, right, that we're an academic lab, we were very focused on translation. We had to make this very cost effective. That means we had to rely on manufacturing processes that were established, that were scalable. We ended up settling on sort of additive forms of manufacturing, as I'll describe, which will allow us ultimately to, including the cost of goods, make these things for just a few pennies. So again, allowing us to borderline compete with the cost of a tablet. And as I sort of emphasized up to now, we really wanted something that could be distributed like a pharmaceutical, even like a topical cream, where basically the doses are built into the device in a way that's sort of transparent, and you just get the right device and you apply it, and it will deliver a single dose, and it would therefore be prescribed just like pharmacotherapy. All right, so that was the idea, and then half a decade later, we had our first functional prototypes. So this is the basic idea. There's a pouch, right? There's a plastic enclosure, and everything is inside the pouch, including a printed battery pack. So that's both the power source and the regulator, all right? The only thing that's outside of the pouch are these two interface hydrogels, like these two little sticky pads. And then you stick it to the body, right? And what that does is it completes a circuit, right? Where now the body is connected to the device, and it is the completing of the circuit that actually activates the device. That's why there's no on-off switch, right? It's pressing it on, activates the device, and all the functionality, therefore, comes from what's inside the pouch itself. The idea is very simple. The implementation was rather complicated. So these pouches, though they can be even 0.5 millimeter thick, at any point can include between 10 to 16 layers of different materials that have been added. You can imagine sort of printing them layer by layer, and you actually have a very complicated three-dimensional electrochemical structure with different elements that I'm going to be describing in a second. I mentioned we really wanted something that was scalable, right? It's one thing to have something that, everyone takes a skilled PhD student 3 weeks to assemble. It's another thing, right, that in theory could be printed in a sheet-to-sheet or even a roll-to-roll manner in a second. And so these were the processes that we sort of adapted for this process. What you need to imagine is that you start with a substrate, and then you're just printing, you're adding each layer. And so the structure is being formed, right, as you're printing these different layers, resulting in the final composition. This includes things like the active electrochemical elements itself. So this is another cartoon of the process. So as I mentioned, the thing is sort of like a pouch. I want you to imagine that initially you start with a flat sheet, and you're printing onto that sheet, and that's what's being illustrated here, and you're making the different layers. And then in the final stage, right, then you cut out the sheet, and then the final stage, you fold it in onto itself, right? And so it's in that final folding where you have sort of the top components and the bottom components that you actually create the final structure, right? And then the only thing that has to be stuck on the outside is the hydrogel element. So this was part of the design process that we came through to realize this. A lot more details, of course. At the core of these are batteries. There's a lot that goes into the design of the batteries. I'll see the materials, their geometry as well, and how they are stacked. Each battery will provide, based on this chemistry, only about 1.5 volts. That is not enough voltage for most applications that we're interested in. And so what you do is you have to add batteries in series, right? Now, this has to be done in a very artful way, since they're being stacked in this planar way. But the basic idea is that you're now combining multiple batteries. And these can be homogeneous, meaning they're all the same kind of batteries, or they can be inhomogeneous, meaning each battery is actually different. And plays a different role in the regulation of the output. Each battery is a sealed compartment, and so we had to do a lot of work on things like glue, right, and adhesives to figure out how to formulate it. As a byproduct of the batteries being active, they can gas, so we had to create a microscopic venting system because it's not hard. It's not like a hard shell like a regular battery, so they would otherwise sort of bubble up. So we had to create this microscopic one-way venting system. We had to invent this technology for the through holes, so the pouch is only perforated in one spot, and that's where these hydrogels are stuck. But the hydrogels need to be connected to the terminals of the batteries, so you need these sort of through holes. So you have to make the holes, you have to fill them with very prescribed metals. We spent a lot of time on conformability. So these devices aren't flexible. I wouldn't call them flexible electronics, but they have to be conformable enough to fit on the part of the body that they're supposed to be able to fit on. And so again, a lot of work was done, including realizing that the batteries are always going to be less flexible than the sort of space between the batteries, so we can design these sort of axis of flexure. And this, again, is all going to be very specific to the design itself. All right, so this notion of controlled stimulation without electronics is very bizarre to neural engineers and electrical engineers. And the reason it's bizarre, and also to people who work in batteries, the reason it's bizarre is you now have the battery pack and you have the body, and they're both rather complicated and nonlinear. So people who work in batteries, right, know that when you connect a battery to any piece of electronics, you need some electronics to control the discharge of the battery. Batteries don't regulate their own discharge, right? A battery doesn't say, I'm going to provide 1 milliamp for 20 minutes, and then I'm going to stop, right? Batteries don't have that capability. And so the electrochemistry of these batteries is well characterized, but how to make them self-regulate is not. Now, the body itself is also a problem because the body isn't just a simple resistor. The body itself is sort of this nonlinear adaptive load. As you put current into the body, you change the impedance of the body, right? And this is why conventional electronics, conventional stimulators all use current control stimulation, which essentially involves feedback. And so now we have these two complicated nonlinear systems, right? And we want to create a situation that when we couple them together, we can provide a very controlled prescribed dose, right? Such as 1 milliamp for 20 minutes, right? Not more and not less. So to do that, we had to essentially develop a new theory underlying the design. I'm going to describe that at a very high level. Our approach is to characterize separately the load and the battery pack, and then finally to simulate their performance together. So what does that mean? And again, all the details are in this preprint. So this first step of characterizing the load, the way we do that is we take just the hydrogels, and we put them on the body. We connect that to a source meter, essentially a constant current source, and we discharge, we stimulate it with fixed levels of current, right? So we'll stimulate you with 1 milliamp for 20 minutes. We'll stimulate you for 2 milliamp for 20 minutes. We'll stimulate you for 3 milliamp for 20 minutes. And we measure the resulting voltage. So this is without a battery pack, right? This is just a hydrogel. We are characterizing you, and this is what you look like, right? There's the different current levels we've tried. There's the time that we're applying you, and there's the voltage. And you can see it's not a simple plane, right? It's sort of bending in a very particular way. That is your body's nonlinear response to the stimulation. All right. Let's leave the body aside for a second, and let's take just the battery pack or the cells themselves. We discharge them under current control as well, right? So this is under galvanic static discharge. So we're taking the battery pack without you there, and we're drawing current in a fixed way, 1 milliamp, 2 milliamp, 3 milliamp, and we're measuring the resulting voltage. It's sort of a measure of the internal impedance, it's more complicated than that, of the battery pack itself. And we get a plot that looks like this. So again, this is just a battery pack. There you have the current. We're discharging different levels over time, and you can see how as we discharge the current, the voltage actually, these battery packs drop, right? It's normal, you're drawing current, you're going to lose voltage on your batteries, but it's a very complicated slope, right? It depends a lot on the details of the electrochemistry. Now it turns out that the two things I've just shown you have the same axes. They're very different, but they both have current, time, right, and voltage. And so what we did is we then overlay these two plots. Let's see if this animates out, right? So there's the plot of you, and there's the plot of the battery pack. And they will intersect at a very particular point, right? So for each time, there'll be associated voltage and current. And then another time, there'll be another voltage and current. So what that line is showing you there is a journey through time. And as you're moving through time, you're passing through specific currents and voltages. It turns out that doing this process yields very reliable predictions of how the battery pack will in fact discharge on the body. I'm going to be honest with you, it did not have to work out this way. Exactly, right, because it didn't have to work out this way, but it did. And so this allowed us then to start using this theory to design packs for specific applications. And the reason we need to do that is these are very complicated to design, they're very complicated to build, complicated to test. We couldn't build 1,000 variations, right, and try them on 1,000 people, right? We had to use computer simulations to optimize first, do computer-driven design, and then, based on what I just showed you, take our best designs, our best trajectory, and only apply those on subjects. And these had to be application-specific. There are 3 applications we initially were very interested in. One is head stimulation, brain stimulation. So this could include TDCS. One was transdermal current-driven drug delivery. All right, this is a technology called iontophoresis. And another one is accelerated wound healing, so using electricity to help wounds heal faster or better. We chose all 3 of these because they're not new. Our goal was not to invent new indications. Our goal was to find existing indications, right, where there was extensive clinical data for efficacy, and yet not a lot of deployment, right? Why not, right? So those are the issues I talked about. And so once you identify these indications, you're able to identify the dose, this is the single per dose, that these devices should put out, and they vary by indication. So in the case of wound healing, you look at the literature, it's less than about 100 micrograms, but you're gonna want it flowing for, quite a bit of time. If you look at something like brain stimulation, much more current, right? But stimulation for a shorter period of time, and iontophoresis, the drug delivery, it's more charge defined, and the application also determines the shape of the device, right? So this is what these devices look like. Oh, let me jump ahead. So this would be a head stimulation device, right? This was the device I showed you earlier. This would be a Band-Aid-like device, right? So they look very different, and so there's very different design inputs leading to very different designs. In the case of the TDCS device, we ended up needing 4 batteries in order to get the required voltage, therapeutic voltage, as well as thinking a lot about the flexure of the device. For the iontophoretic device, instead of using a hydrogel at one location, we essentially used a non-woven felt, so like a sponge, and in that sponge you would insert whatever it is, the molecule that you wanted to inject in, right? So that could be whatever you're interested in, some painkiller, right, a vaccine, whatever you wanted in there, and obviously when you put it on, the current would start, and then the drug would be delivered. For accelerated wound healing, I want you to imagine that you have a little cut on your hand, and the approach was that we have over the cut a conventional non-woven felt, like something soft. The sticky hydrogels go outside, right? So the sticky hydrogels are the interfaces. And the current flows from one of the sticky hydrogels to the other sticky hydrogel, but as it does that, it crosses across the wound, right? And so you have 2 sticky things and one non-sticky thing in the middle, like a Band-Aid, right? It ends up being just like a Band-Aid, right? Except this Band-Aid is energized, okay. And so that's how the current would flow. Once we design these things and build these things, the main thing we need to validate, like the key functional element, is that when we put it on the body, it gives you a single dose at the prescribed level, right? So if it's the Band-Aid version, it needs to provide, right, somewhere between 40 microamps for 2 hours. If it is the brain stimulation version, we needed to first ramp up. It can't go up instantly in this case, right? We needed to ramp up, hit a target level, and then self-deplete after a certain amount of time, right? And this is across multiple people, right, who present very different loads. All right, let me show you some results, specifically with the Band-Aid. This was tested in a rodent model. The nice thing about the rodent model is the rodent-sized device is like the same as the human-sized device, actually, so it's the same form factor. This is, it's a puncture wound, and then the Band-Aid is put across of it. This is just showing the current level that was being passed based on prior preclinical work. We set a target of 300 micrograms for 2 hours. This is just showing you the trace of different applications along with the lighter lines, along as what the average dose looks like. And this is what an exemplary result is. So in the top trace, you see wound healing in this rodent with a regular Band-Aid, right? The non-energized version. And they heal pretty quickly on their own. So this is a full thickness wound. And this is over 14 days. And you can see in this case, by the 14th day in this animal, it's almost, not fully, but it's almost fully healed, right? So that would be the control. In the bottom trace, you see the effect of the energized Band-Aid, right? So this is the Band-Aid that's providing current, and the current's provided for 2 hours. And every day, we put a new Band-Aid on the animal. So every day it gets a new dose. So 14 days, 14 Band-Aids being put on and off, and obviously they're being replaced also in the control animal as well. And you can see even by eye here that there's faster healing in the stimulation case. You can also see by eye that it almost looks like it's healing in sort of sealing in this sort of slit-like pattern. That's actually the direction of the current flow, and that's something we didn't predict ahead of time, but which makes a lot of sense based on the mechanism of healing. And obviously this was repeated in multiple animals and quantified more systematically, and that's in the paper. And so there is a statistically significant increase in the rate of healing. We've also noticed less infections in the stimulation group. And I should say all this wasn't new. All this was in fact expected. There has been, without exaggeration, 60 years, maybe more of work of people using electrical stimulation to accelerate wound healing. Rodent work, clinical work, post-surgery wounds, diabetic wounds. It goes on and on and on and on. Yet where's the deployment? Why is it not in CVS? Why does your doctor not give it to you, right? And again, so the issue we're dealing with here is not reestablishing a scientific principle of can electricity accelerate healing? How does electricity accelerate healing? We're leaning on decades of work in it. We're just trying to give it to you in a very different form factor, right? If your kid has a wound, I don't know if you're going to be that excited to get a device that's this big, make sure it's charged, get the cables, blah, 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 stick one electrode here, one here, I don't know, put it in their backpack, right? And tell them, you know, not to run, whatever it is, right? As opposed to just getting something like this, right? Okay, whatever, 20% faster healing, right? 50% less chance of infection, why not? Right? And so that's really the vision. Another application we're very interested in, because it's something I've worked on a long time with conventional stuff, is transcranial direct current stimulation. So this would be wearable disposable TDCS. This is the actual device itself here. We don't need to make them transparent, but we just make them transparent so you can see the batteries inside. You can see the 4 batteries here. It's put on the head. It has an automatic ramp up. It's stimulation over a certain duration, and then a ramp down. So that's something we had to engineer. You can't have the sudden transients. All that is being controlled, by the way, by the electrochemistry, right? There's no timing electronics, there's no ramping electronics, and so on. We considered 2 different current intensities. One is more conventional, and we've also actually started to explore higher currents, 5 milliamp up to 10 milliamp. This montage is one that is studied extensively for things like depression treatment, which is why we focused on it. Again, auto-regulation of discharge without electronics. This is what the device looks like on the head. As soon as it's placed, it starts to discharge. Now, how do we know that it's doing what it's supposed to do? Well, there's no electronics in here to give us that information, so we just need to have little tabs, and we add a source meter on top of it. So that's how we actually are able to validate that it's doing what it's supposed to do. I mentioned we have 2 target currents that we're interested in. And a long story short is, across multiple subjects, the devices were doing what they were prescribed to do. They were ramping up, hitting a target current, and then ramping down. Now, if you're looking very closely, of course, it's not exactly, right? It's not exactly 2.5, right? Sometimes it's 3, sometimes it's 1.5. That is true. So we don't have the same level of precision as you do with electronics. But I think the variability that we see here is much less than the variability that you would see from other sources. Certainly when you swallow a pill, right? The amount that gets to your brain is not the same, right? Depending on who you are. So we don't have to have a standard for perfection. For those of you who do work with TDCS and are maybe noticing this higher current, for a few decades, TDCS has not been able to go above 2-3 milliamps because of tolerability concerns. We discovered that the hydrogels that we were using allow much more tolerated stimulation, up to 5, 6, 7 milliamps. This is a great example of students not listening to the professor when he tells them explicitly, don't try this because it's not going to work, and the professor is wrong. And just very briefly for, just going to skip this. So it's as tolerable as regular TDCS, just because the hydrogels, turns out, have a great capacity to pass current through the skin. And so this opens up new dimensions for dosing as well. Okay, so let me wrap up here. So looking at a use case, this is conventional TDCS versus wearable disposable TDCS. Conventional TDCS, you have the equipment, you have the disposables, you have to set it up every time, you got to program it, whatever. There's an app, there's the Bluetooth, you got to still throw out all the stuff. Well, in case of wearable disposable TDCS, you would just grab your 10 doses. You could carry it around however is convenient, and use it when is convenient. And so I think we set, I'm just jumping through this for time, I think we've sort of set a goal to develop a brain stimulation electrotherapy device that could be used much like pharmacotherapy, could be used like drugs. And I think we've done it. There's still obviously a long way to go as far as deployment, and ongoing clinical trials will happen in the fall, but I'm very excited for the future. Thank you.
Okay, thank you so much for this wonderful talk. So I think we have time for 1 or 2 quick questions about this immediate work before we move on to the, obviously you can ask bigger questions at the end of the session.
Thank you very much for the talk. I don't think it's on. Test. Thank you very much for the talk. It was really interesting. My name is Anthony. I'm a high school student, and my question is, without an interface for the user to see if the device is on, how do you envisage the customer knowing if the device is working well and if the battery is discharging properly?
That's a really great question. It's interesting. So with this new frontier of technology, all these great questions come up like that. One way is you can feel it. Right? So you can actually, depending on the dose we put in, you can actually feel a very light amount of tingling. We can actually intentionally make the hydrogels less good to increase that level of tingling. So if we wanted to make, let's say, a Band-Aid, normally you wouldn't feel 100 micrograms, but we could engineer the devices so you feel a little bit of tingling, and so that's how you sort of know it's working. But also maybe draw the line with a drug. I mean, when you swallow a pill, you don't know that it's not sugar, right? And so there is a little bit of faith on the behalf of the user, right? Or cream, right? You don't know that it's working. So I guess it would fall more into that category. You're correct. You would lose a visual feedback. We could add it, of course. We could try to add some sort of LED or something on it, but then we would lose our other benefits. It's a great question.
Thank you.
Thanks. That was super interesting. My question is really related to the neuromodulation, the brain stimulation, because there's a certain amount of precision that you need as to where you're stimulating it in the brain relative to maybe your other 2 applications. So how do you foresee solving that problem of stimulating the temporal cortex versus frontal, versus where you place that on the brain?
Yeah. So here again, our goal is not to reinvent the wheel. So existing applications for TDCS often just use 2 electrodes. The resulting current flow is focalized to the front of the head, right? But it's not focalized to a specific gyri. And there have been multiple RCTs that have shown that this approach is associated with minimal side effects, itching, and is effective. And so we are piggybacking on that work, saying, okay, that work says that this is apparently focal enough to produce the right risk-benefit profile. And so we're doing the same as well. Of course, there's things like high definition and other things which allow you to be more focal, and you could try to engineer that into these bands as well. So I guess the short answer is we're not, at this point, our goal is not to be, we're actually trying to not be innovative as far as dose, and we're just trying, for the most part, we're just trying to provide what has already been provided out there in this other form factor. It's a good question.
Okay, thank you so much again. Yeah, let's thank Dr. Bikson again. I think the next step would be that I'd like to ask all speakers of the symposium to come back on stage, and we have our general Q&A. And at the same time, I encourage everybody to formulate your questions and come to the microphone here and ask questions to all the speakers that might be relevant, maybe for one or two or to everyone. That would be great.
Okay, hello, this is Hasan Ayaz from Drexel University, and I have a question for Dr. Marom Bikson. Well, thank you for sharing your impressive solution for the scalability challenge of neurotechnologies. And you addressed both cost, miniaturization, and user interface simplification. So do you think one of these dimensions is more important than the other? And if you further go deep down in one, you can improve the scalability potential. Like, for example, if you further miniaturize the system, would it help? It looks like you still have potential on all these. And the follow-up on that is, can you comment on the regulatory pathway that you picked for this? Because that is the efficacy demonstration of the overall system. Thank you.
It's a big question. I'll give you a quick answer. We're not worried about miniaturization right now. I mean, the active elements are in the milligram range. And so actually the size is almost all the enclosure. So that's one of all the problems we've had. That's not one of the problems you've had. I mean, the other issue, you really talk about things like cost and scalability. And I think this is really the difference between, let's say, academic-centered research and really thinking about translation. If you're at that laboratory scale, it's okay to make 100 a month, or maybe okay to make 1,000 a month. In this case, scalability means, making 1 million a month. You know what I mean? And so it's a completely different scale of process. And same thing with cost. Again, if you're satisfied staying at that academic stage, frankly, you can make each one for $100, right? As long as the grants allow it. While when you're thinking about translation, obviously, the pressure becomes very, very high to get to sort of a Band-Aid or a pill level. So there's no simple answer to that other than to say that, really with the translation, the pressures are high. And as far as regulatory, as long as we can keep this in the non-significant risk range, we can make that process much simpler. And I didn't talk about it, but a lot of the engineering went into risk management. These things are, I say borderline edible, and then someone says eat it. But they certainly won't, the electrochemistry prevents an excessive discharge.
While others think about their questions, I have another question actually I think it applies pretty much to all of you. Like, when I think about bringing this, from the lab into the real world, I think what's interesting to me is also like the long-term effects, of stimulation or BCI usage. And I find it hard to distinguish, like, for example, things like gait in the course of disease probably like get worse anyways, right? So how can you distinguish like things if the kind of the treatment effect decreases over time? Do you know? So the question is like, do you know if that happens? Does the effect decrease over time? And how do you know if that's because of the disease getting worse, or is that something that is more like maybe technology, like, I don't know, impedances getting worse, or the system kind of habituates or adapts or something like that?
I can take a stab. So for the gait-related research, these patients have been implanted for over 2-3 years now. And actually, Ken did analysis in looking at stability of the biomarkers over that time span. And for most of the subjects, it has been surprisingly stable over a year or so in terms of the biomarker from when he first tried to whittle it down versus the latest visit. So I think, I guess maybe that's advantage of less invasive in terms of like non-intracortical, right? With ECOG and local field potential, the signal seems to be pretty stable. But you're right, in terms of disease course, with disease progression, like their dynamics are going to change. Yeah, we haven't found a great way of addressing it besides, periodically checking in to see how well these, classifiers are performing.
Yeah, for brain-computer interfaces, for intracortical electrodes, until the last year or two, all of the human work had used that device that I showed you, the Utah array. And so it's a rigid device. It's mechanically mismatched from the brain, and the materials tend to degrade over time. So I think that's every device that's implanted, the signal quality does degrade, and then therefore performance is lost. There are now other devices that are actually in clinical trials led by companies that are using flexible materials or targeting different structures, like even trying to use intravascular approaches. All of these have trade-offs. I think it depends, like what level of information do you actually need to get from the brain? If you just need an on-off signal, you probably don't need an intracortical electrode. If you need that level of movement, though, there needs to be advances in the materials and design in order to preserve that longevity. And just, as the other speakers were saying, as for people who have neurodegenerative conditions, certainly that's you're fighting an additional factor where the actual information that you're able to extract does degrade over time as well.
That was a long one. Hi. Mine's quite a high-level question, but when we think about scalability and neurotechnology, how do we think about how we engage people with this technology? So we kind of tell people, we're going to do this kind of work with your brain, you're implanting deep brain stimulation, for example, and we're thinking about trying to do this over millions of people. How do we engage? For example, I work with a lab in Sheffield who do DBS, and they've had neurologists that have never seen someone from a BME group, minoritized ethnic group. How do we engage people like that in this technology? How can we work with different groups to make them understand what we're doing and how we can integrate it wider into society, and how we're doing that moving forward?
Yeah, I mean, I do think it's a big challenge, right? I was really struck in your talk of there've been therapies that have existed that people just don't adopt, right? Because the actual process of getting them is too challenging. And so that's certainly where we are with brain-computer interfaces. Only a handful of surgeons in the world know how to implant these devices right now. It takes a team of people to calibrate them and to keep them operational. Even in our case, when we do have a home system that people can use, some participants have caregivers who are really willing to learn how to attach the device and kind of get things set up from day to day, and others don't. And so I know that a lot of groups and companies are working to develop systems that are kind of always on, that the person themselves can use, that requires wireless technology that does exist now. But I think that there's still going to be some hesitancy, right? The benefit needs to outweigh kind of the risks and challenges associated with doing that in order for people to start to adopt it. I would say that implanted technology does have lots of applications, just deep brain stimulation, even pacemakers for a heart. Sometimes you do need to implant in the body to deliver the amount of function that you are promising, right? A wearable would be great for a BCI, but we don't have access to the level of movement-related information that we need to restore upper limb function, to restore speech. And so you do need an implanted technology, but it needs to be something that can be scaled. The surgical approaches need to change, the cost needs to be lower, and it needs to work all of the time. Like reliability and accuracy requirements for assistive technology is very high, and so we have a pretty high bar that we need to go after.
Yeah, and just from like the movement disorder perspective, DBS is one of the most underpenetrated and underutilized technology. There's so much class 1 evidence that it works great, yet only probably 10 to 20% of the patients who qualify actually end up getting it. But I think this is a power, in terms of scalability, like a power of industry collaboration, right? Medtronic now, their state-of-the-art generator that everyone's putting in now has this sensing function, and you can activate it. And adaptive DBS, a few months ago was FDA approved. So this is possible. I think as it generates more interest, and there are a lot of patients who advocate and who want it, hopefully that will spread this knowledge and interest beyond just a few academic centers that are really invested in the technology and understanding it. And now with a lot of the data that the companies have access to, like NeuroPace and Medtronic, it would be a fantastic industry, academia, research collaborative to understand what the signals are doing on a much larger, like hundreds of patients, hundreds of thousands of patient scale.
I mean, it's a hard question what you asked. I think it's sort of what maybe partially what this conference is about. I mean, there's this question of how much do we need to do educationally, how much do we need to do versus advocacy, or is the tech just not good enough? You know what I mean? And so it goes back to the engineers and the people who are pioneering translation. And maybe the second part of it is true as well. I mean, DBS has been around for a long time, but maybe it's been begging for adaptive technology, and that's what it's needed. And same thing, with prosthesis, the fact that we need, to integrate these other harder technologies, these sensory things. So sort of blame the technology, not just the education. The both are clearly important.
All right. Another question that I had on my mind, it's probably, again, relevant probably to all of you, but I certainly saw it very clearly in Dr. Bikson's talk. Like, you need to think probably a lot about factors that go beyond just research, right? Thinking about like bringing this technology to like actual people, or to the clinic, or to everyday life. So, and again, that's probably a huge question, but like, could you say just a few words about like what are the actual like the biggest challenges that you're facing, like bringing this technology, these approaches from the laboratory to the actual like people to use it, or clinicians to use it, or people at home?
I mean, I think one thing, and I think this is true for all of us, is we need to recognize, you have the sort of the academic front, and then there's industry. And I don't think anything that ultimately, when you get to patients, ultimately labs don't build this technology. We like to think, okay, we're academics, we do everything, but we don't build medical devices. We're not qualified, regulatory-wise qualified to bring medical devices. So that's one thing. I think it's you have your industry here. I know these other speakers must work very closely with the companies that they deploy. Maybe that's something to think in mind about how to be more open to those academic industry collaborations, and figure out how to do that ultimately, to the best benefit for patients.
Yeah, I completely agree with that. On the device side, that's not going to be my role of trying to improve the device and making a device that gets implanted perfectly every time and gives you the best signal quality. But I think what we've seen, kind of as I've already said, is the reliability requirements are high. And so a lot of what we're trying to do in the lab is try to push the science, try to understand behavior, see how that's impacted by sensory feedback. But the first products need to be something that works every single time in every single situation, no matter if the person is sleepy or on a different medication or in a different environment. And so I don't think that first generation of products is going to have the degree of function that you see from different academic research labs, but it's going to be based on those scientific findings and collaborating with industry to deliver something that is really robust and performs well.
Yeah, and the scalability is a huge question, and I think like Medtronic is running into that now with the deployment of adaptive. Like, how do we actually use it in patients? And even a center like as accomplished and well experienced as UCSF, even our group is having challenges in like who's the best candidate? How do we track this? And it involves a lot of patient engagement. It's not like, we implant the device, we turn it on, it's like a light switch. It takes, we're distilling like months and years of data intense collection with like postdoc students, analyzing it to figure out what works for that patient. But if you're trying to do it on a mass scale, that's really challenging. So, and then Medtronic, they have to now hire, PhD neuroscientists to kind of figure out that problem as well. So yeah, I think it goes both sides.
Okay, perfect. Thank you so much for all your answers, for all the wonderful insights. I think with that we're gonna.
Sorry, can I?
Yeah. Do you have another question? Yeah, please. Go ahead.
Okay. Yeah, hi. I'm Melanie from the University of Oldenburg. Yeah, my question is mainly directed towards Dr. Wang, but maybe Dr. Collinger and Bikson, you also have something to add. So I was very intrigued by the swimming study because I also did EEG during swimming, so yeah, that was something nice to see. And yeah, so of course I'm also interested in swimming, and so I was wondering whether it might be a therapy approach as well to like take out the gravity from walking and to like either walking in the water environment or also just swimming to try to improve like motor function in patients with movement disorders, because I think there are also studies reporting that some Parkinson patients can like cycle when they're like on a spinning bike. Yeah, if we take those motor function that are still somehow still present in those patients and try to train them, that maybe functions like walking might also be improved over time?
Yeah, that's definitely a really interesting idea. I think I think there are physical therapy rehabilitation programs using like aquatherapy for patients not only with Parkinson's but with like injury and neurological diseases in terms of motor function recovery. And I think one of the things, actually I trained a physical therapist in my lab who completed a PhD. It just, we do all these therapies and there's no neurological basis. So the effects are often short-lived and it's not targeted. So I think one of the main motivation is like if we can understand what specifically within the brain circuit is disabled and whether we can train that circuit to prevent freezing, preventing falls, that would be a lot more fruitful, I think, to couple like rehabilitation with an understanding of targeted neuroscience approach.
Thank you again. All right, I think with that we're gonna finish that session. Thanks all our speakers again very much.